Good morning to everyone in the room, and good afternoon, good evening to the online audience. I am thrilled to be here today to talk about some bioanalytical issues found from the recent FDA BIMO inspections and remote regulatory assessments, RA. Here is my FDA disclaimer. The learning objectives of my talk today include to get familiar with the relevant US laws and regulations, to understand the basis for demonstrating bioequivalence, BE, and also to make you aware of some bioanalytical issues from recent FDA BIMO inspections and RAs. And here is the outline of my talk today. In 1984, the Hatch-Waxman amendments of the Federal Food, Drug, and Cosmetic Act introduced the new abbreviated approval pathways of marketing applications. Specifically, a 505(b)(2) application is an NDA containing full reports of investigations of safety and effectiveness, where at least some of the required information comes from studies not conducted by or for the applicant, and for which the applicant has not obtained the right of reference or use. Meanwhile, a 505J application is an ANDA for a drug product that is a duplicate of a previously approved drug product, and generally must contain information to show that the proposed generic product is pharmaceutical equivalent and bioequivalent to the reference listed drug, RD. Additionally, the Code of Federal Regulations, CFR, Title 21, also includes relevant sections. For example, I listed here, there are Part 312, the Investigation on New Drug Application, Part 314.54, Part 314.92, as well as Part 320, Bioavailability and Bioequivalence. Here, I am showing you all the table of content for CFR 21, Part 320, Bioavailability and Bioequivalence, which lays out the legal foundations for requirements of bioavailability and bioequivalence studies. You may find the details online if interested. In Part 320, the basis of demonstrating BE is defined. Specifically, two drug products, test and reference listed drugs, RLD, are considered as bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternative. And the rate and extent of absorption do not show a significant difference when administered at the same molar dose of the active moiety under similar experimental conditions, either single dose or multiple dose. And if the rate of absorption is different, the difference would be intentional and reflected in labeling. And the extent of absorption should not show a significant difference between the test and the reference, or the RLD. For drug products that are not intended to be absorbed into the bloodstream, BE may be demonstrated by using scientifically valid methods that are expected to detect a significant difference between the test and the reference products in safety and therapeutic effect. In addition, FDA also publishes guidance documents to describe the agency's current thinking on certain topics. The guidances do not establish legally enforceable responsibilities and should be reviewed only as recommendations. For example, here I'm showing you the guidance M10 Bioanalytical Method Validation and Study Sample Analysis published in November of 2022. This guidance describes recommendations of the bioanalytical methods used to measure concentration of chemical and/or biological drugs in biological matrices obtained in all phases of clinical trials in regulatory submissions, including comparative BA or BE studies. And the table of content displayed here is showing some expected critical elements during method validation, including selectivity, specificity, matrix effect, calibration curve and range, accuracy and precision, carryover, dilution integrity, and stability. Last but not least, the FDA also has the Bioresearch Monitoring Compliance Program, BIMO. It's a comprehensive program of on-site inspections and data audits designed to monitor all aspects of the conduct and the reporting of FDA-regulated research. The BIMO was established to assure the quality and integrity of data submitted to the agency, FDA, in support of marketing applications, as well as to provide protections of the rights and the welfare of human subjects and animals involved in FDA research, FDA-regulated research. Among several BIMO compliance programs, the program 7348-004 describes the coverage of a typical in vivo BABE analytical inspection, like I'm showing you here. Again, you may find it online if you're interested for more details. Now, let's switch gear, and I'm going to talk a few case studies. The first case study, this one, is a BE study with PK endpoint submitted under ANDA. The drug product was a small molecule and also an endogenous compound. The study was designed as a randomized two-treatment, two-sequence, 4-period, crossover, single-dose, and full replicate trial. And the bioanalysis of the samples was conducted using LC-MS. During the FDA inspection or RA, objectionable conditions were observed. Specifically, there were higher pre-dose analyte concentrations observed in the subjects that's receiving test products compared to the reference listed drugs. Not all the subjects, but only some of them. And also the pre-dose analyte concentrations did not exhibit expected physiological variability in the subjects. The firm being inspected could not provide adequate explanation or justification of the above observation. No discrepancy was identified during sample analysis. However, like I mentioned before, the firm could not offer any explanation or justification would support the observed pre-dose discrepancies between the test and the reference products in this subsets of the subjects. Therefore, OSIS concluded that the analytical from the clinical study are not reliable for agency review. Case study number 2. This was an in vivo BA study with PK endpoint submitted under an NDA. The product was a large molecule and was analyzed using ligand binding assay, LBA. During the inspection or the RA, objectionable conditions were observed. Specifically, the assay actually did not demonstrate sufficient accuracy to measure the analyte concentrations. Specifically, the calibration standards and quality control samples, they were not prepared using a reference standards of known purity. Nonetheless, the calibration standards were used to back calculate, assign, and report specific analyte concentration for the subject samples. This subject's concentrations were later on used to evaluate and support the endpoint in the clinical studies. OSIS concluded that this observation impacts the study data reliability because the site did not use analyte reference material of known purity to prepare the calibration standards and QC samples. Thus, it is impossible to calculate and report accurate analyte concentration of the study subject samples. However, the percent change from baseline analyte data should be reliable because the method actually demonstrated adequate precision when measuring instrument signals from the QC samples. That is, the method consistently measured higher signals in high QC samples and low signals in low QC samples with adequate reproducibility. OSIS inspectors also verified that the instrument was properly maintained and calibrated according to the firm's SOP and CLIA requirements. Thus, the method showed adequate precision and the percent change from the baseline analyte data were reliable. All right, now it's time for some challenge questions. Challenge question number 1, true or false? You must follow all the recommendations listed in the FDA's guidance for industry for bioanalytical method validation and sample analysis. A, true, B, false. Can I get a show of hands? Who's picking A, true? Half, maybe 0. Okay. Option B, oh, sorry, I gave up. Yeah, so the answer is false. Like I mentioned before, earlier in my talk, the FDA guidance documents, they are not legally enforceable, and they should be viewed as the recommendations only, and it reflects the agency's current thinking on certain topics. All right, challenge question number 2. Which type of evidence may be used to establish bioequivalence BE? A, comparative PK study. B, comparative PD. C, comparative clinical endpoint. D, in vitro in vivo correlation, IVIVC. Or E, all of the above. Can you guys just shout out what you would choose? Perfect. So all those about 4 study types could be used as evidence to demonstrate bioequivalence. And here are some references that I mentioned during my talk. And if you are interested, you should be able to find them on the FDA websites. With that, I would like to thank my OSIS colleagues and management support to make this presentation possible. Thank you so much for your attention, and I'm happy to answer any questions you may have during the panel discussion. Thank you. All right, now let's welcome Michael to the stage, and he's going to talk about MHRA's perspective in their findings from recent inspections. Welcome, Michael.
Hello again. So as Cynthia said, I'm going to take a UK slant and talk about bioanalytical observations and findings from recent inspections. Again, just to bring this slide back up, we'll talk about something different. So concentrating on the very well put together graph, 3D image you can see on your right, distribution of findings. We'll go through not in a lot of detail, and you will pick up some of the clinical ones from Emma's presentation later on. But there you can see the number of critical findings, major findings, and other findings that we've raised since 2019. The aim of this presentation is slightly different. We have a slightly mixed audience. We've got pharmacovigilance later. We've got bioequivalence in the morning, both clinical and analytical. So the findings that we talk about here have been deliberately selected to try and give you food for thought, and with the idea in mind that if you take care of these, you won't end up in the critical finding list. Just in terms of, I said there'll be 2 audience participations, but one of them includes a prize. For this one, by show of hands, can I ask those that are directly connected with regulated bioanalysis in the room? If you're online, please feel free to raise your hand, I just can't see you. So anyone directly connected with regulatory bioanalysis? Hannah, thank you for your hand. Okay, that's great. There is no problem, we will walk through all of these together. So as I said, here's a breakdown. You will find this again as Emma's slides. So in the red or orange box, depending on what you can see, that is what we've classed as our clinical findings. And Emma Whale will go through that in session number 3 later today. What we are going to talk about is the major findings in the blue box. So as you can see there, method validation, data integrity controls, and clinical sample analysis. As I said, the intention here is to stop those findings turning into these findings. It has been very deliberately ordered that you can see method validation, data integrity, which is the, if you like, big brother or sister of data integrity controls, and clinical sample analysis. So as I said, we've got 3 examples to talk you through. Just in terms to give those context, some of you may have experienced an MHRA inspection in the past. We classify our findings in those categories. So a critical finding is where a significant departure from the principles of GCP have been identified, which either put the subject, clinical trial subject's well-being at risk, or the integrity of the data cannot be relied upon. A major is something that, if action is not taken immediately, will result in a critical finding. So if you like, it's a warning shot. So just to take that down, we'll break this into the method validation example I've selected is around stability. The clinical sample analysis will be on run acceptance, so how you decide whether or not an analytical run is valid. And in terms of data integrity controls, we are going to talk about dynamic data. So what I want you to think about as we go through these examples is making effective use of regulatory guidance. Cynthia's talked about that. Guidance is exactly there. It is not a thou shall, but if you're going to do something different, you need to think about it, document it, and go through. The idea of transparency and how crucial that is, particularly where you're going to do something innovative or novel. We want to be able to reconstruct your thinking, your processes. We don't want to rely on your colleagues' memory when you've won the lottery. And the last part there is data security and control. It is unfortunate that a number of inspections in my 9 years at the agency have resulted in negative outcomes, not necessarily because the method doesn't work, not necessarily because they weren't diligent, but because the system which you had acquired the information to leaked information was open to abuse. And it's one of the things that is incredibly frustrating as an inspector. So, to go into method validation, on the picture you can see there is a lovely -80 freezer with some samples. What I want to think about is long-term stability is the assessment of stressing the samples by putting them in to a freezer designed to mimic subject samples during the clinical trial and pulling them out at defined points. If you think about it like a lottery ticket and waiting for the draw, you put them in hope. And then as each number comes up during the draw, you get a little bit closer to the jackpot. You get to decide your subject samples. The bit I want to talk about, and what we've seen on inspection, is that a number of laboratories have failed to analyse the prepared samples, stability samples, immediately after generation. So if you like, you're your lab scientist, you've got your lab coat on, you've got all of your samples in front, you are spiking your low quality control samples and high quality control samples ready to freeze. We'll go to the guidance part in a second, but at that stage, you have a huge amount of variables that can affect the outcome of the assay. Whether the analyst's pipetting technique, if that's what you're doing, is on point that day, whether the tip has got a manufacturer's defect, whether or not there is something else at play within the method. The idea of doing the analysis up front is to give you as clear an indication of what you can see later on, that you knew it was all right when it left there. Now, again, I'm going to share some ICHM 10 references, and Cynthia's links are there because I tried to hyperlink this and it didn't work. So what I've done is taken the direct wording from ICHM 10. So this is specifically section 3.2.8, handily titled stability. And the part in bold is unambiguous. It talks about aliquots of low and high QCs are analysed at time 0. Going back to my hamstrung lottery example, it is the one thing that you can do to improve your chances of the jackpot. It gives you future information about it was okay at the time. And it's a missed example, sorry, it's a missed opportunity if you don't take it. So, what we'll do there, the text you can see currently is a slimmed down version of the deficiency. So laboratory procedures did not contain a requirement to analyse stability samples immediately following preparation. Therefore, There is no assurance that stability samples met acceptance criteria prior to storage. Now, this has a number of outcomes as we work through it. But at this stage, long-term stability is something that you do right at the start. It's done in method validation so that you can lay your samples down because you have to wait for them to age. It is not something that if you get to the end of your clinical trial that you can jump back. You have lost that opportunity. So what can you do if this is your facility? And this is a very common finding that we're seeing currently. We've presented on it in the past, and I'm hoping in talking about it today, I can stop writing that finding. So the first thing is to recognize that you have a problem. Look at whether or not you have time zero samples. And I will find most of you that work with laboratory scientists, they are quite thrifty. They will turn one experimental procedure into many. So you might find, although it wasn't designed to be time zero, that your low and high QCs might actually have been analysed by luck. And some of the cases we find on inspection, that's exactly what's happened. So that's the first case scenario, what I would call the best outcome. The next part is if you don't have that, then you're starting to look at the reliability of the long-term stability, how it's deteriorating over time, the percentage of nominal for your low and high QCs. And that's a really difficult thing to do because what you're doing is replacing an experiment with a human's opinion. And as all inspectors will tell you, we would much rather have data. Now, it's not the end there though. You might have seen that your time point 1 to time point 5 all come out beautifully in line with nominal, plus or minus 6%. So well within acceptance criteria. But you might also find that time point 1 passed and time point 3 passed, but time point 2 didn't. And you're left in that horrible situation where you can't say it was okay when it went in. And that situation crops up more than you would think. So again, try to incorporate this into your procedures as early as possible. If you don't have it, it's not the end of the world, but you should start documenting that now. The next one we're going to talk about is clinical sample analysis. And again, this is run acceptance criteria. So I know a number of people in the room don't work directly in regulated bioanalysis, but this is the part when you get to the end of the assay, you've got your calibration curve, you've got your QCs, and what you're doing is looking to see if you can trust the unknown concentrations that you've got as a result. So those are incredibly well designed. They're to take away any subjectivity or ambiguity that it shouldn't be my decision or your decision. It should be that's what we've agreed up front and that's what we're doing. So what are we seeing on inspection? The inclusion of company-specific criteria. So we've mentioned ICH M10, FDA have their guidance as well. In addition to that, previous to that, you have the European Medicines Agency's Bioanalytical Method Validation Guidance 2012. And what those talk about is a defined list. So you're looking for assay accuracy precision, you're looking specifically at the calibration curve, whether or not you can regress lines against it, you're looking at your extraction method through QCs, you're gathering a wealth of information and then putting that back in. So that's one thing. If you as a company decide we want the best data, we want our gold-plated analytical results that are irrefutable to a regulator, and we're going to insist on this. That is, as we've said, a possibility. Document it in your SOPs. The second part in bullet point 2 is that these additional criteria are not always applied consistently. This is where you end up in a problem with the inspector. Because as we said, bioequivalents are slightly different to some trials. The design is relatively simple, 2-period crossover, open label. The outcome for a comparative PK assay is very well defined. You know what a pass-fail is. Most regulators, all regulators I would say, will be able to tell you that, a confidence interval of 85 to 110, or 90 to 110. So now you've got this real fear that bias, whether it's conscious or unconscious, can be introduced by the decisions that you make. So something that would normally have been regulatory guidance, unambiguous acceptance criteria, suddenly becomes, that's just an SOP deviation. And that's the dangerous place to put an analyst or a scientist in, which might mean that they have to work a double shift at the weekend. So take the subjectivity out. What I would say, if we go into, again, it wouldn't be a regulator's presentation without where we're taking this from. So I've slimmed this down from ICH M10. But again, it's really clear. It talks about rejected Cal and QC calibration curve. This is a specific example for a comparative PK assay where the company were including a criteria. If I go back. Nope, apologies. Where the company had included their own criteria that if they lost the lower or the higher calibration curve, then the run would be rejected. Regulatory guidance incorporates the reason why you might not want to do that, so long as your QCs are covered within that run. And for the example that we're talking about here, we saw a selective use of this criteria being applied. So, again, on the right-hand side, from there you can see the deficiency that we would raise. So laboratory procedures were found to contain issues with respect to analytical run acceptance criteria where requirements were not in accordance with guidance. Consequently, and this is the part where we're talking about bias, the policy for analytical run acceptance would have otherwise been, as a consequence, the policy for analytical runs which would have otherwise been considered acceptable were repeated and the original analysis not considered for pharmacokinetic output at the end of the trial. Now again, in terms of considerations, one thing that I would challenge whether you're working with a bioanalytical lab or a study monitor is one, are these additions essential? Are we talking about gold plating or is there a scientific reason for why this needs to be in your comparative BE study? And if they can't answer that question, my suggestion is that you ask them to work to the guidance. You take away any form of guidance. They might have a very well-documented rationale. Again, when we've seen this, where we've seen that it being applied inconsistently, again, one thing that we've struggled with is that documented rationale. Why are you doing it? Why is it in place? What can be done? Okay. So, the last example that I want to talk about is data integrity controls, which seems like I haven't stopped talking about it since 2018, and the use of dynamic data. So what are we seeing? And again, this is in the major category. We have examples where this has failed and we're talking about criticals, but this is the precursor to there. This is where you have the opportunity to affect change. So what are we seeing on inspection? We're continuing to see static data being used for review of bioanalytical data, which has been acquired in a dynamic format. Just skipping ahead to the MHRA's guidance and a plug, other guidances are available, including the FDA's. Raw data must permit full reconstruction of the activities. Where this has been captured in a dynamic state and generated electronically, paper copies cannot be considered as raw data. What I'm going to do is take you through an example, and this is where the prize and audience participation comes in. What we've got here in the picture is a chromatogram, and it's been used for stability assessment. It is generated in a system with well-defined and what I would call broadly GCP and GXP data integrity controls intact. If we go slightly closer, let's do audience participation. This study was from a comparative PK study where you had LCMS, MS, but not to mislead you, but this chromatogram better shows my example, but it is not from an LCMS, MS. Anyone in the room want to offer a potential what method this might have been from, what machine? And I can offer you a carbonated Scottish soft drink. Feel free to shout out in the room. Again, online, I'm really sorry. Not LCMS, MS, anything else that would be in that chromatogram generating sphere before I open it up to my colleagues on the right-hand side. Okay, that is fine. Anyone from the inspectorate, whether UK FDA or Health Canada, want to offer? No, I'm running ahead of time. No. Other soft drinks are available. Okay, so this again, we're talking about dynamic data. What this company were doing was printing out the chromatograms and presenting it for review. Now, again, everybody in the room, I imagine, has some experience in quality assurance. There's something that sticks out about this, something that doesn't appear right. That's a lovely gap down the bottom. So the chromatogram printout was modified prior to the review stage. And what I would say here is the removal of the acquisition date and time sticks out like a sore thumb when you highlight it. Now, the reason this is a problem is we're talking about stability. It's not the chromatogram itself. The chromatogram actually was acquired very well. It was within acceptance criteria. It can be used. but the timing, the metadata when you acquired that injection is incredibly important, because what it does is show that I can rely on your data. What they'd done is forgot to take the sample at the right time. So the scientist, in some misguided attempt, said that's fine. And they waited that out and submitted it to QA and QC, like all the way through until we turned up on inspection. And we are used to looking at gaps, so we found this. So in the interest of time, I'm not going to read the deficiency. But again, it's there. You can see that the dynamic state is crucial to it. One, you've spent time, money, generating a system that can generate all of your analytical data with audit trails. It's got a Watchman directory that moves it out of a modifiable format. You might have Active Directory to link it directly to an individual. There is no way of changing it. But yet you print it out and you let them submit it. So in terms of considerations for this, I want you to think about why would that be necessary. One, it's easier. I can take a piece of paper, I can go and sit at my desk. It takes a bit more thought to understand why you might want to then go to the system, go to the lab, have the yellow light, have the noise. That's not a conducive environment for review, but it's not the reason to not do it. The removal of the opportunities is only part of the puzzle. And I think I've gone through 3 examples, and what I would say is, if you remember nothing else, it's the second part of this. Creating a system that is tightly secured and tracked by audit trails, the modifiable data is moved by Watchman directory to a safe, secure location, the archive is all intact as one piece. Humans are relatively resourceful, and again, in 9 years, I've found that people have found a way if they want to do this. So what you need to do is look to achieve the right environment where they can make mistakes. Not doing the stability sample at the correct time is not the end of the world. Removing the acquisition date and time is. That is the one that breaks regulators' trust. We then talk about where else have the modifications come. And going back to that, that is a very difficult crack to fill. So, I want to pull this up. The idea of the talk was to try and take you from that wiggly blue box to remove them completely and avoid getting to this grey-black box. What I want to leave you with is a short summary. Look at regulatory guidance. It is our current thinking. If you do have a good reason for doing something different, make sure you document it. Your decision-making and the documentation around that is crucial. We don't want to rely on hearsay when we turn up to do an inspection. Exercising control of your data is important. Don't let all of your hard work with the CSV, the periodic reviews go to waste. And act before issues escalate. And with that, I'd like to thank you for your attention and pass back to Sean.